I'll be talking about the evolution of polygyny, or the multiple queen social form in the red reported fire ants. Also, one of the main questions in evolutionary biology is we try to understand how a certain phenotype, how a certain traits arise. But now we know that it's from the interaction of its underlying genetic basis and its environmental context. So a lot of people are interested in finding what the key genes are and what's the expression pattern like. So I am personally more interested in the organism itself on a phenotypic level. So given what we know about the genes and about the ecological content, how does selection shape an organism into what it is? So when I was applying for grad school, I knew I wanted to study insects. But I think to myself, what is the most interesting system in insects? Right? It has to be social insects, where you have this reproductive division of labor of different castes. You have thousands of individuals in the colony, but effectively they function and reproduce as one single superorganism. It's just fascinating, right? And this is a quote from E.O. Wilson that my advisor Ken really likes, that social behavior comprises a set of phenotypes furthest removed from DNA. So any animal behavior, even a single one, could be really hard to decode and understand, not to mention this complex social behavior, especially those in a social insect colony, where all these network and interactions, all these individuals collectively perform a certain task and to ensure survival of the colony. You wouldn't really expect them to have a single or a simple genetic basis, but that is actually kind of what's happening in fire ants, which I will tell you about in a bit. But so we study ants, and a lot of people in the many entomology departments in university, they're really interested in ants. So why ants, right? And they're absolutely everywhere. Wherever you go, you see ants. That means they're highly abundant. It means they're ecologically very successful, and they're very high both in terms of the biomass and diversity. And so in Georgia, like one of the prominent species is the red reported fire ants. If you live in the southern states, I'm pretty sure you heard of them, or at least or you see them maybe. They are an invasive species from Argentina, came into the U.S. around 1930s, and they have now established almost all the southern states, as well as many other temperate regions and subtropical regions in the world. So they are advanced eusocial, meaning that they are the highest, they have this highest level of sociality, that the queen is reproductive, but the workers are completely sterile. The workers cannot lay eggs, cannot reproduce, they don't have functional ovaries. And one reason we study them is that they are easy to collect and keep in the lab, because they're just so abundant, they're everywhere. So they're used as a model species for the study of social evolution in ants. So one feature that makes them interesting, and also something that our lab focuses on, is the queen number variation in the colony. So a fire ant colony could have either a single reproductive queen, we call them monogyn form, or they could have multiple reproductive queens at the same time in the colony, or we call them polygyn form. And gyne means reproductive female. So these are really two distinct kind of social forms. You either have one or you have many queens. So then the question is why? Like, why does colony have one queen and why have so many? So in some sense, this transition from monogeny to polygyny can be viewed as analogous of the transition from solitary to social lifestyle itself. Or more generally, it can be seen as one of the major transitions in evolution, where you have you start with one simple entity and then a collection or a group of this entity becoming a new level of individual. So we're hoping that by studying the evolution of polygyny, we can understand something about social evolution itself. And what makes them more interesting is that it's not just the queen number that vary between these two social forms. There's actually a whole syndrome of various phenotypic traits that's associated with it. Like in polygyny form, their nests are much closely distributed to each other. They are less aggressive to other polygyny colonies, and the mode of colony founding is different, which I'll talk about in a bit. And the size of the queen and the workers are also quite different. And it turns out that this complex syndrome has rather a simple genetic basis, or at least in terms of the number of or like this gene element, a wild type haplotype, we call him the S-b supergene. It is formed by chromosomal inversions, and it contains about 500 genes. So although it's quite large, but the entire supergene inherits as one single Mendelian element. And the figure here where there were fluorescent markers that attach to the gene. In the wild type, you see that the green comes first, and in the supergene allele, you see the blue comes first. So how does this supergene regulate the social form variation, right? In monogyn form, you don't find this supergene, it's absent. But in polygyny forms, all the queens and a big portion, about an average 65% of workers bear this as little b supergene. From now on, I'll use little b to represent the allele that has the supergene. And one thing you should know that is little b, this kind of genotype almost doesn't exist in nature. And it's because the supergene has accumulated quite a few deleterious mutations, and it's become recessive lethal at some point. And the same story goes for males. When males that have the supergene is also pretty weak. And in Hymenoptera and ants, males are haploid and females are diploid. So they have this haploid diploid sex determination system. So queens mate almost exclusively to big B males. So they don't, if they mate with a little B male, they are very unsatisfied. They mate again with a big female. So you don't find little b genotype, essentially. And we know that it's actually the workers that determine what kind of queens to have in a colony. If in an ant colony, right, if the colony is queen right, in monogyn colony, as long as the mother queen is present, they will not tolerate any additional queen. That's why they are monogyn. That's why they only allow one single reproductive queen. And of course, if the colony is queenless, they would accept any queens of any conspecific queens. But in a polygyny colony, no matter how many queens you already have in a colony, as long as you give them a queen that have the supergene, the colony will happily accept an additional queen to it. And in nature, this happens usually after a mating flight event, where usually it's after a rained and on a good sunny day the next day. So the winged females and males, they go out and mate in the sky, and then afterwards they land and they remove their wings. So monogyn queens, those that don't have the supergene, they will usually attempt to independently start their own colony. But the queens that have the supergene would tend to join an existing polygyny colony. And that is how a polygyny colony grow their size and expand the territory. So how does the worker know, right? How can the worker tell that this is a conspecific queen and you have supergene or not. One thing you should know about ants is that they communicate primarily using chemical signals. Imagine you're a worker, that this is an ant colony, and then you have all these larvae of different age and life stages to take care of, and then you have to recognize a lot of things, right? But then in reality, they live underground. It's completely dark. They can't see nothing, so they have to rely on a really good sense of smell to tell where everybody is and to be able to perform appropriate brood care. And each member of the colony has a unique particular chemical profile associated with it. Particular chemical means that the chemicals on the insect's exoskeleton on their surface. So, and so it's been found that the particular chemicals it varies regarding the caste and sex and age, and even depends on what kind of task you're performing in colony. It all varies. So everyone has a unique profile that's identifiable to you. And you can see on the right here, the profile of a reproductive queen is quite different from that of a worker right here. So where does my research comes in? I'm interested in two aspects of this discrimination behavior. One is I want to know what's the key chemical compounds that are conveying this supergene message. And the other project of mine, which I'll talk about in the second half, will look at behavioral patterns of the workers and how they, how little bee bearing workers convince the whole colony to accept additional little bee queens. So a study that's kind of like a basis for my first project, that they found that using paper dummies that dosed with cuticular extract, they show that the workers of fire ants can discriminate the genotype of the supergene based on just cuticular chemicals, just like how they discriminate a live queen. But unfortunately, we still don't know what exactly are those chemicals, and that's what we're trying to find out. So on the cuticle of queen, you have various different kind of compounds. We have first the CHCs. The CHC stands for cuticular hydrocarbons. These are the type of molecules that are long-trained with only carbon and hydrogen molecule. They are usually function as a waxy layer on the surface of insects to prevent desiccation, but they're also co-opted to use as semiochemicals in a lot of insects. And these cuticular hydrocarbons can be divided into saturated and unsaturated, depends on if they have double bonds in their molecule. And besides that, you have a bunch of polar compounds. These are the compounds that have a higher polarity than the cuticular hydrocarbons. And the major components in the polar compounds are a group of chemicals called the piperidines. And the piperidines are they are structurally very similar to the molecule in pepper plants that gives the hot and spicy taste. And pepper plants are in the genus Piper, so that hence the name. And piperidines are also the kind of molecule in the venom of fire ant workers that makes you painful when they sting you. And there are also other kind of polar compounds, like in a minor amounts, like some aldehyde, ethanol, that. And if you look at the pattern of abundance of unsaturated cuticular hydrocarbons, you see that their abundance really increase, or I just say they have a lot of them on the queens that with the S-little supergene, but they're effectively absent in queens without the supergene. So we think that, oh, this strongly suggests that maybe some of these unsaturated cuticular hydrocarbons is conveying the presence of the supergene. And And if you look at the pattern of abundance in some of the polar compounds, especially in piperidines, you see that they're very abundant in reproductive queens, and they're less abundant in non-reproductive queens. So these are the queens that have not yet made it. They still have their wings, and they are effectively absent in workers. So this tells us that reproductive queens has it, and you're less sexually mature, you don't have it. So perhaps piperidines is a good candidate for the queen cast signal that tells the worker that I am a queen cast, I have really good fertility condition, depends on how much piperidines I have. So these are the two ongoing hypotheses that we based on our project on. Right, so then what do we do, right? We collected colonies of both forms around Athens mostly. And actually in the picture here, these are a bunch of polygyny colonies. And the picture in the middle, these are one big monogyn colony. You can see that the polygyny colonies are much closer to each other, and the monogyn ones are they have a much higher peak. They're further away from each other. And these are some video footage of how we collect the ants. So we just shovel up like the entire soil, and then we dump it into a buckets, and then we carry the buckets to the lab, and then we use what's called a drip flotation methods to slowly drip water into the buckets to make it flood. So then, and then the workers would aggregate and form a kind of a raft, and then that's how we collect the content of the colony. How do we extract that from the soil? And then drink some water. So using this method, we can just extract only the ants and the larvae and the adults, and just leave the soil behind, because we don't. Because this way it will be easy for us to manipulate the ants when we want to do experiments. And then we keep the colonies in all these trays in the lab, right? All right, so and then what do we actually do in terms of assays to try to identify the key compounds? So we take about 3 grams, about 5,000 polygyny workers, and then we give them a new tray, a temporary nest, and give them water tube and food, and then we leave them queenless for 24 hours, at which point they would, if you give them a queen-like object, they're going to take it into the nest. So after 24 hours of queenless, we give them 2 dummies, and one is the MIR dummy, which the MIR stand for monogyn incipient reproductive. These are the queens that just shed their wings at the absence of the mother queens. So they just start to activate their ovarian development. So they are going to smell like a queen, but the MIR extract do not have the supergene signals in them because they're from monogyn colonies, right? And then at the same time, we give them a treatment dummy. And the treatment is dosed with different fractions of reproductive polygyny queen crude extract. And the dummy is just a piece of filter paper that has a size and weight similar to an actual queen. So the idea is that if the treatment has effective supergene pheromones compounds, the workers would strongly prefer the treatment over the MIR. We have this matrix that's developed to score the preference towards the treatment over the MIR. So if you just give them the MIR, which smells kind of like smells like a queen, but does not have the supergene signal, they would retrieve it. But if you give them 2 dummies at the same time, they're going to prefer the treatment over the MIR if the treatment is effective. And this is what it would look like if you give them an effective treatment dummy that's dosed with reproductive polygyny queen crude extract. And in this kind of assays, we call them the positive reference, which is like giving us a baseline of how they should perform, right? If the chemical contains effective pheromone compounds. You can see that the workers are usually quickly get excited and they're going to gather around the dummy and they would develop this trail to pull the dummy as if it's a queen, and then they're going to pull it and rescue it into the nest. This usually happens within 10 to 15 minutes. And there you go, almost there. Right. So this is what we look at, right? So if the score from a kind of a series of treatment assay do not differ from the positive reference, then we say, oh, this treatment compound ineffectively replicates the queen supergene pheromone that mediate this kind of S-Phylloby supergene discrimination. So let's look at the result. Right? This is a distribution of the score of different assays with different treatments. The first is the positive reference. The first group here is that their score is pretty good. They're close to the top highest score possible. And if you give them a combination of the unsaturated cuticular hydrocarbons with either the MIR or the polar fraction from the polygyny queen, they are effective. The scores are pretty, they're just, it's not different from the positive reference. But if you remove the unsaturated hydrocarbons, like you can see here, the yellow dots, they become ineffective. And also, if you give them only the unsaturated hydrocarbons, but not MIR or the polar, they also become ineffective. So that is clearly stressing that the unsaturated hydrocarbons are really an essential group in this pheromone gland. Right, but it's unsaturated CHCs itself is not enough. You also need to pair it with the queen Cas signals. But surprisingly, the queen signal is not in the piperidines molecules, which we hypothesized. Right? So again, the green group is the positive reference, and if you give them unsaturated CHCs with either the MIR or polar, they are effective. But if you give them unsaturated CHCs plus just the piperidines, but not the other kind of polar compounds, as you can see in this group right here, they become ineffective. So that is a surprising find for us. So that is telling us that the queen Cas signal does not lie in the piperidines. It's actually in polar compounds that is not piperidines. So we don't know what those compounds are. And to recap, right, we're pretty happy to find and to confirm that the unsaturated cuticular hydrocarbons indeed likely signal the presence of the supergene queens. Interestingly, it's the polar compounds that are not piperidines that signal the queen Cas status. And another thing is we also try to further divide and fractionate the unsaturated hydrocarbons to find out what are the key compounds in it. And turns out you really need a really complex blend in it. That it's not just one or two compounds, it's a whole bunch of them. So that's like the first project. And now I'm going to transition to the second part, which is the behavior aspect. So in a social colony, they're often used as a model to study what's called collective decision making. As you can imagine, this group of thousands of individuals often need to collectively perform certain tasks, like where to forage, and where to locate a new home, as it's necessary. And a lot of times, you cannot afford to have every single individual in a colony to participate in a decision, right? And in the case of nest hunting in Thamnophorus acorn ants, and as also in honeybee swarm, they send out a few number of scout individuals. The scout will seek out a new home, and they would debate among themselves, and they would determine the new location for the entire colony. So we really have the opinion of the minority that and can influence the decision of the entire colony. And there was a similar story in fire ants that only 10 or 15% of Philippi workers is sufficient to induce polygyny for the entire colony. And this was found in 2 previous studies where they cross-foster either queens or larvae brood of the alternate social forms into a colony, and then they create a colony fragment with various portion of the little bee workers. And they found that as long as you have more than, say, 10-15% of workers that carry the supergene, it will become a polygyny form. You become a polygyny. So, like, how does that happen, right? That's what we are trying to find out. What's the key factors that influence this collective decision making? And so we designed a different kind of behavior assay. Similarly, we'll give them a temporary nest, a new tray, and then we'll put in 400 workers, of which 300 of them will be monogyn and 100 of them will be polygyny. And that gives it 18% of little bee workers in an assay units. That is a little bit higher than the critical threshold that convert the social form. And a control unit does not have polygyny workers. It just have monogyn from monogyn colony. So we don't all just put in a bunch of workers together at the same time because they will fight, right? And the monogyn workers will likely kill the polygyns. And so we put in the monogyn worker first, and then we leave them in there for 24 hours, and then we add the polygyny workers in a cup, and then there was a metal screen in the lid of the cup that prevents them from getting out. So that way, the two types of workers can interact on the screen, and they can antulate each other. They can like smell each other, get used to the scent of the other type of workers, so that reduce the aggression level when we release the queen. Right? So that give them another 24 hours, and then we release the queen. Oh, not the queen. Release the workers. And at this point, we give them 2 polygyny queens, and to ask them basically if you would accept the polygyny queens or not. So in this version of the assay, the polygyny workers are 2 hours. We release them 2 hours before we give them the queens, and then we videotape all the interactions, and we'll see what kind of interactions go on. And this is what you would see on the behavior assay. On the left, it's the control assay, where all the workers are monogyn workers, and they're like pinning down and attacking the introduced polygyny queens. Well, on the right, the bigger workers are the monogyn ones, and the smaller workers, like this one, are polygyny. And in this case, even though they have really a small number of polygyny workers, they become receptive to the polygyny queens, and they would develop a trail and retrieve the queen into the nest. So that's what you would see in this 2-hour kind of assay. And then subsequently, we developed different version of the assay to reduce the contact and interaction between the 2 worker type. So what you saw was the 2-hour version of the assay 8, where we released the polygyny workers 2 hours before the introduction of the queens. And in another version, we released them only 10 minutes before introducing the polygyny queens. That means they only have 10 minutes of free interaction before they encounter the queen. Well, yet in another version, we do not release the polygyny workers at all, so that they can So the polygyny workers are always in the cup. They never freely interact with the monogyny workers, or they also cannot directly contact the queen. And in another version, in the SAC, we put a double screen layer on the cup. In this case, the two type of workers cannot even intimate each other at all. They cannot physically touch each other at all. And also their workers are not released. So from A to C, the contact and interaction between two types of workers gradually decrease, and we'll see at what point do they stop accepting the queens. Right? And this is what we see in terms of the acceptance. And no matter if you look at the acceptance percentage by counting the total number of queens accepted or the number of units that accepted any queens, you see a really clear and strong pattern of that when you have more contact and interaction, then you will have a higher level of acceptance. But there is a significant jump from B to A, as you can see here, from about 26% to 70%. And the difference between the version the assay A and assay B is that in assay A, the polygyny workers are released from the cup, and in assay B, the workers are not released in the cup. Right? So in B, the workers do not freely interact, and then also the polygyny workers cannot touch the queen at all. So that is telling us potentially this full body interaction between the monogyny worker and polygyny worker can be really critical, and that is suggesting perhaps there are some kind of chemical cues from the polygyny workers that would be acquired by the monogyny workers that let them somehow be more perceptive to the supergene cue of the queen. And also that is suggesting the interaction between polygyny workers and the queen could also be important. Maybe when the polygyny workers are contacting the queens, they are releasing some form of signal to tell the other workers, oh wow, I like this queen and I want to retrieve them. I want to rescue her into the nest. So at this point, we cannot distinguish which one of these is potentially more important. We'll have to design new experiments to kind of dissect that. And we're thinking about a different version of the assay where we set up the assay like the other ones, give them 300 monogyny workers, 100 polygyny workers, but we will restrict the polygyny workers from contacting the queens, but they can contact the monogyny workers. And we do that by like tying a little wire on the polygyny workers, and then we put the queen into a petri dish, and there will be a hole on the petri dish so that the monogyny workers can get in, the polygyny workers cannot get in at all. So in that sense, we can tell whether the interaction between the polygyny workers and the queen is more important than the interaction between these two types of workers. So that is pretty much all I want to talk about today. See that I am a little bit ahead of time, but yeah, that concludes my talk, and I guess I'll take any questions at this point.
Great talk, Horace. So any questions? If you want to ask questions, just unmute yourself and just speak.
Right.
Unmute yourself.
Hi, Horace. You did a great job. It's a good talk.
Yeah.
I have two questions for you.
Yeah.
One, how many polygynes have you seen in a nest or colony at one time?
You mean Polygyny queens?
Yes, how many queens?
It very varies, and they could have hundreds of queens in a
Oh,
colony.
okay. Yeah.
Yeah, yeah.
Wow.
They usually have a lot. You don't see just they use in the wild. It's you kick open a mount, you can usually find a dozen of queens, and when you collect them and you move them into the tray, they see actually they have 100, 200, even sometimes even more than that.
Oh.
So yeah, there's really no limit in terms of how many queens they can have in a colony.
Okay, so I think Phalaenopsis invicta has like super colonies, right? So the super colonies have to have polygyny queens in order to be a super colony because they need to be able to work together. But then super colonies can be separate. Do you like have any idea like if that has anything to do with the super gene, or like how, you know, they would how they become like super colonies? Like, you think that
Right,
they just learn to accept each other?
So the notion of super colony is essentially this group of colonies in the same local area that uh, are tolerant of each other, right? They're basically accepting a neighboring colony as a sister colony, and that's the case in polygyny fire ants that they're very they're not aggressive to each other, and yeah, that is indeed due to the presence of the supergene, I would say. So somehow the presence of supergene make them uh, more perceptive of each other as long as you have the supergene. That's why we call them. They have this kind of wind beer effect, and but they but if you keep a two polygyny colony in the lab environment, and if you give them different diets and they're separate for a really long time, they can potentially become aggressive to each other if they're segregated and long enough, basically.
So their like environments are different. So their colonies wouldn't be
Right.
close together anymore.
Right. So as long as they live in the same area, they're eating relatively the same diets, and they live in the same kind of soil, same climate environments, they are tolerant of each other essentially. So that's why we you move a queen or you move a worker from one polygyny to another polygyny, be no problem. They would accept it.
All right, thank you.
Yeah.
Might I add one thing to Sabrina's question and Horace's answer? It's true that the polygyny workers are very tolerant of other polygyny workers from different nests in the same area. But what's really interesting is that it's been found that there's an association between queen number and worker relatedness in a nest, and also between queen number and worker size. So the more queens you get, the lower relatedness the workers in that nest, and the smaller the workers. And it's very difficult to understand how such correlations could arise if these nests were actually freely exchanging queens and workers among each other. So although they seem to be tolerant of each other, I think that the nests are somewhat contained in the sense there's not wholesale willy-nilly migration of queens and workers from one nest to another.
Yeah, because that might have like an evolutionary effect almost, I guess, like with like problems with like the genes that are maintained if they were more willy-nilly about it.
Well, I would point out that these are recently introduced invasive populations. So all bets are off in terms of questions like that having to do with the evolutionary significance.
Okay.
Horace has probably got a better answer for that than I.
What do you mean by that exactly?
I just guess like if two colonies, like, you know, if the more the colonies that are related to each other, I guess the more likely they decrease in like the variation of their genome, right? Is that related? No, am I wrong?
I guess you could say that. So this absolutely supergene, right? It's almost like a parasite in the genome of our ants. It itself inherits this, a Mendelian element, and it's like its own thing, right? But at the same time, it cannot be homozygous with its own copy, and it has to come with a wild type haplotype, right? So even though they don't recombine, the supergene don't recombine with the wild type, there is still the rest of the genome that's important. That that is like the majority of the genome. That, yeah, I don't know if that kind of answer your question, I guess.
It does. Thank you.
Cool. Any other questions? I have a relatively, I would say, maybe easy question. So you show that the polygy colony tend to live close to each other. The monogy are usually further away. So I guess it has to do with that the polygy are more friendly. Monogy are that kind of attack each other. You also show that the monogy has a bigger nest. Why they do? I think they are smaller, right? Why do they need a bigger nest?
Right. So monogyn colonies usually have a higher, bigger soil mount. If you look at the outside, the surface, and presumably, according to Ken, that's due to maybe the concentration of the queen pheromone in the colony. In monogyn, you only have one queen, right? The pheromone is concentrated at one spot. So all the workers kind of work around her and kind of like build mounts to a point to higher and higher. When polygynes, we have so many queens, right? They're distributed in a wide area. Mm-hmm. So when the workers are trying to build mounts, they're less likely to concentrate at one direction, right? They're going to expand more. So it's wider, flatter. That's perhaps why the shape of the mounts kind of differ.
Yeah. Good. Thank you.
Yeah.
So if there's no other question, I'm going to randomly put people into a breakout room, and you are welcome to stay as long as you want. I'm going to keep this keep the meeting on all the time until people are gone. Okay. So okay.